There is a creature outside your window right now. You cannot see it, you cannot hear it, but it can hear you, your breathing, the rustle of your clothes, the soft percussion of your footsteps on the floor. It is painting a three-dimensional map of the world in sound, updating that map 100,000 times per second, and navigating through darkness so complete that your eyes would be useless. It weighs less than a handful of coins. It has been doing this for 50 million years. Tonight, we go into the dark with the bat. Not the bat of Halloween decorations, not the creature of folklore and fear, the real one. The one that is by almost any measurable standard one of the most sophisticated animals to ever evolve on this planet. Over the next 100 facts, we will dismantle everything you thought you knew and replace it with something far stranger and far more astonishing. Bats are the only mammals on Earth capable of true powered flight. Not gliding, not the elegant cheat of a flying squirrel stretching a membrane and trusting that gravity does the rest. True flight sustained, steerable, generated entirely by muscle and wing. Birds do it. Insects do it. And among all the mammals that have ever lived, in over 220 million years of mammalian evolution, only one lineage cracked the code: the bat. To understand what that means, consider what flight actually requires: a complete restructuring of bone density, metabolism, muscle architecture, and respiratory capacity. The bat's body didn't just add wings, it rebuilt itself from the skeleton outward. Every system reorganized around the demands of staying airborne. Scientists still debate exactly how this transition happened, because the fossil record offers only glimpses, but what emerged was an animal so aerodynamically refined that aerospace engineers still study its wing geometry today. The word echolocation dramatically undersells what bats actually do. It sounds clean, clinical, like a dial you turn up and down. The reality is almost incomprehensible in its speed. Some bat species emit ultrasonic pulses at rates approaching 100,000 per second during the final moments of an insect pursuit, a phase researchers call the terminal buzz. At that rate, the bat is receiving and processing acoustic information faster than the human nervous system can register a single visual frame. Each returning echo carries data on distance, velocity, size, texture, and trajectory, all decoded, all acted on in the span of a wing beat. And the bat does this continuously in total darkness while flying at speed, while dozens of other bats are doing the same thing nearby. The processing power required is staggering, and it all fits inside a skull you could hold between two fingers. A bat's call can exceed 100 decibels at the source. To put that in perspective, a jackhammer at close range reaches roughly 100 decibels. A bat mid-hunt is essentially screaming. And yet the human ear hears nothing. Bat calls operate in the ultrasonic range, typically between 20,000 and over 100,000 Hz frequencies that sit entirely above the ceiling of human hearing. This is not a small gap. The highest note a young human can perceive sits around 20,000 Hz. Some bats call at over 100,000. That is like having a conversation conducted entirely above the range of a dog whistle in a room where the acoustics change every millisecond. The bat lives in a sonic world that is simply inaccessible to us. We share the same air, the same night, the same sky, and experience entirely different realities within it. A bat hears its own echo returning in under 1 millisecond and adjusts course mid-flight. 1 millisecond. That is 1,000th of a second. In that time, the bat has received distance information, velocity data, and target dimensions, compared them against its internal model of the environment, and issued corrective instructions to its wings. The neural pathway from ear to brain to muscle is so compressed, so optimized, that the response is essentially reflexive. The sensory motor loop, from echo reception to wing adjustment, operates faster than most human voluntary movements can even be initiated. Every wing beat is a decision. Every decision happens faster than thought. The horseshoe bat's nose is not decorative. That strange, convoluted, almost grotesque structure, the horseshoe-shaped nose leaf that makes these bats look like something from a fever dream, is a precision acoustic instrument. Horseshoe bats emit their calls through their nostrils rather than their mouths, and the nose leaf acts as a sound-focusing dish, shaping the outgoing pulse into a tight directional beam. Different folds and ridges within the structure influence different frequency components of the call, adjusting directionality, spread, and focus in ways that shift as the bat moves its nose. Think of it less like a nose and more like a satellite dish that the bat can aim with the muscles of its face. The structure varies significantly between species, each variant tuned to the specific acoustic demands of that bat's hunting environment. Form follows function here with ruthless precision. Nothing on that face is accidental. Some bats can detect an object the width of a human hair using sound alone. A human hair is roughly 70 micrometers in diameter. That is not a target, that is a suggestion of a target. And yet the greater horseshoe bat, in controlled laboratory conditions, has demonstrated the ability to detect a wire of that diameter suspended across a flight corridor, using only reflected sound. In the dark. What this means in practical terms is that a bat's echolocation achieves a spatial resolution that sonar systems engineered for submarines, built with decades of research, billions in funding, still cannot match at equivalent range. The bat solved this problem 50 million years ago using cartilage and neural tissue and never filed a patent. Echolocation is precise enough that bats distinguish between two insects flying centimeters apart in total darkness. This is not target detection. This is target discrimination. The bat is not simply locating objects, it is identifying them, separating them, and selecting between them all from the returning geometry of a sound wave. In cluttered environments, forests, reed beds, the interior of caves, where dozens of objects occupy the same acoustic space simultaneously, bats parse individual echoes from the overlapping chaos, with a selectivity that remains only partially understood. Some researchers believe bats use the slight differences in echo timing, measured in microseconds between objects, to build a three-dimensional separation in their acoustic image. Others point to frequency-specific filtering. Perhaps both. The honest answer is that the full mechanism is still being mapped, and each new study tends to reveal another layer of complexity that the previous model hadn't anticipated. The bat brain dedicates a disproportionately large region to processing sound proportionally larger than the human visual cortex. In humans, roughly 30% of the cortex is devoted to processing visual information. Vision is our dominant sense, and our brain reflects that investment. In bats, the auditory cortex is similarly dominant, and in some species so elaborately organized that researchers have identified distinct subregions tuned to specific acoustic parameters. One region decoding distance, another velocity, another texture, each tuned to a single dimension of the same returning echo. It is not better hearing. It is a different kind of mind, shaped entirely around sound. Bats shift their call frequency mid-hunt to avoid jamming by other bats, a real-time acoustic negotiation. When multiple bats hunt the same airspace, their echolocation calls overlap. An echo returning from another bat's call is indistinguishable from your own unless you can tell them apart. Bats solve this by adjusting their call frequency when they detect a neighbor operating at the same pitch, a behavior researchers call the jamming avoidance response. It is cooperative in outcome, competitive in origin. Each bat shifts its frequency to carve out its own private acoustic channel, a negotiation conducted in microseconds invisibly in a medium humans cannot even perceive. The same principle is used in wireless communication engineering frequency division to prevent signal interference. The bat invented it. Evolution published the solution 50 million years before the first radio patent. The frog-eating bat can identify a frog species by its mating call alone and knows which ones are poisonous. This is not echolocation. This is passive acoustic intelligence. The Tungara frog, and others like it, call at night to attract mates, and in doing so broadcast their location, their species, and their approximate size to any bat listening. The fringe-lipped bat Trachops cirrhosis has learned to decode that broadcast. It hunts by eavesdropping. More remarkably, it has learned to distinguish between the calls of toxic frogs and edible ones, avoiding the poisonous and targeting the palatable. When researchers played recordings of unfamiliar frog calls to these bats, the bats could generalize, applying learned acoustic rules to novel species they had never encountered. That is not instinct. That is something closer to inference. The frog-eating bat is not just a predator, it is a listener, and it learned somewhere along the way that not everything singing in the dark is safe to eat. The pallid bat hunts scorpions by listening to their footsteps on sand. No echolocation, no active sonar pulse bouncing off a target. The pallid bat, Anthrazus pallidus, folds its sonar away entirely when it hunts on the ground and switches to passive listening. Enormous ears angled forward, scanning the desert floor for the faint mechanical sound of an arachnid walking. A scorpion moving across loose sand produces a sound so subtle that sensitive laboratory microphones can barely register it at distance. The pallid bat hears it from meters away, drops silently from a low perch and strikes. It is largely immune to scorpion venom, a biochemical adaptation researchers are still characterizing. What strikes scientists most about this behavior is the flexibility it reveals. This bat does not have one hunting mode. It has several, and it selects between them based on context, environment, and prey type. That kind of behavioral versatility in an animal this small suggests a cognitive adaptability far beyond what the word instinct can contain. Some moths evolved ultrasonic ears specifically to detect bat calls, an arms race that has been running for 50 million years. The bat did not hunt an undefended world. From nearly the beginning of bat echolocation, insects began evolving countermeasures. Noctuid moths, among others, developed tympanic ears tuned precisely to the frequency range of bat calls. When a moth detects an approaching bat, it executes an evasive flight maneuver, a hard spiral dive toward the ground, or a series of erratic loops designed to break the bat's acoustic lock. The precision of this response is extraordinary. Moths can detect bat calls from distances of 40 meters or more, giving them enough time to react before the bat is close enough to resolve them clearly on sonar. It is a sensory arms race, written in anatomy, each escalation by one side met over generations, by a counter-escalation by the other. The bat gets better. The moth gets better. Neither side wins. The negotiation simply continues in the dark, every night in every forest on Earth. Certain tiger moths evolved an acoustic counterweapon. They emit their own ultrasonic clicks to jam bat sonar mid-attack. This is the escalation beyond evasion. Rather than simply fleeing, some arc-tired moths fight back acoustically. When a bat locks onto a tiger moth and enters the terminal buzz, that final high-speed barrage of pulses just before the strike, the moth fires back. It produces rapid ultrasonic clicks from specialized structures called tymbals, flooding the bat's acoustic field with interference at precisely the moment when accurate resolution matters most. For years, researchers debated whether this was jamming, warning coloration in acoustic form, or simple startling. Recent studies suggest it may be all three simultaneously. The clicks disrupt sonar, they advertise toxicity, and they startle the bat enough to break the attack sequence. The bat mid-strike receives a wall of noise instead of a clean echo and misses. The moth, unharmed, continues into the night. The moth did not study acoustics. It simply did not want to die. And 50 million years of not wanting to die turns out to be a remarkably effective research program. The greater horseshoe bat compensates for the Doppler effect in real time, a feat that stumped acoustic engineers for decades. When a bat flies toward a target, the echoes return at a slightly higher frequency than the emitted call, compressed by the relative motion between bat and prey. This is the Doppler shift. If the bat did not account for it, its auditory system would be receiving a distorted signal like a radio station drifting off frequency. The greater horseshoe bat solves this by continuously adjusting its outgoing call frequency downward, pre-compensating for the expected shift so that the returning echo lands precisely on the frequency its auditory system is tuned to receive. It recalibrates this adjustment in real time as its flight speed changes as the target moves, as angles shift. The first time engineers fully understood what the bat was doing, the reaction in the research community was something close to disbelief. The bat had independently solved a problem that took human sonar engineering decades to address, and it solved it neurologically without instruments at flight speed in the dark. Bats can distinguish the texture of a surface from its echo, smooth versus rough, without touching it. An echo returning from a smooth surface, carries different spectral characteristics than one returning from a rough surface. The amplitude, the frequency spread, the micro-timing variations in the return all differ based on surface geometry. Bats exploit these differences to categorize objects acoustically. In laboratory experiments, bats trained to distinguish between smooth and textured discs transferred that learning to novel objects, applying the acoustic category to new shapes they had never encountered. This means bats are not simply detecting objects. They are building a sensory vocabulary for surface properties, texture, density, orientation derived entirely from the physics of sound reflection. For a creature that lives in darkness, this is not a secondary ability. It is how bats understand the material world. Touch for them happens at a distance. In a cave housing millions of bats, each individual recognizes its own call returning through a wall of noise. Bracken Cave at peak occupancy contains 15 million animals, all of them echolocating. The acoustic environment inside that cave during emergence is not merely loud, it is by any reasonable measure chaos. Millions of calls filling the same air, bouncing off the same stone, arriving at every ear simultaneously. And yet each bat pulls its own echo from that chaos. How? Part of the answer lies in call individuality. Each bat's voice carries a personal signature, subtle enough to distinguish from thousands of others. Part lies in the brain's ability to suppress what doesn't match. Scientists still debate the full mechanism. What they agree on is this: no human-built system has replicated it at this scale. The cave solves the problem every evening, 15 million times over, without a single error. The wing membrane of a bat contains embedded sensory cells that detect airflow changes, turning the entire wing into a touch-sensing organ. This was not understood until relatively recently. For decades, the bat wing was studied almost exclusively as an aerodynamic structure shape, surface area, membrane elasticity. Then researchers discovered that the wing skin is densely populated with Merkel cells and hair follicles connected to sensory neurons, the same cellular machinery that gives human fingertips their sensitivity. These sensors detect changes in airflow across the wing surface in real time, feeding information directly into the bat's flight control system. A sudden stall, a gust, a shift in air pressure, the bat feels it across the entire span of both wings simultaneously and adjusts. The wing is not just a surface for generating lift. It is a distributed sensory array, gathering aerodynamic data on a scale and resolution that no aircraft instrumentation system currently matches. The bat flies by feel across every centimeter of its wingspan all at once. Bats can detect prey moving underwater, sensing the ripple from below and striking from above. Several bat species in the genus Noctilio, the bulldog bats, have refined echolocation to operate at the water surface interface. They fly low over still water-emitting calls that reflect differently off the flat surface than off a protruding object. When a fish rises close to the surface or breaks it, the echo signature changes, and the bat detects that change from 3 meters above. In flight, in darkness, it then rakes its elongated hind feet, equipped with curved claws, through the water at precisely the right coordinates. The timing required from echo detection to foot deployment is measured in milliseconds. In some regions, these bats are significant predators of small fish, operating entirely nocturnally, in an ecological role most people would never attribute to a bat. The boundary between air and water, which seems categorical to us, is just another surface for the bat to read. Scientists still debate whether bat echolocation evolved once or independently multiple times. There are two major groups of bats: the echolocating microbats and the largely non-echolocating flying foxes and fruit bats. For years, the assumption was that echolocation evolved once in the common ancestor and was then lost in the fruit bat lineage. But genetic and morphological analyses have produced conflicting signals. Some evidence suggests that the two groups are not as closely related as their shared wing structure implies, and that echolocation may have evolved independently in each lineage. If true, that means the same extraordinarily complex sensory system, with all its neural architecture, its call production hardware, its auditory cortex specialization emerged twice separately, from different starting points. Convergent evolution of this sophistication would be remarkable enough to reshape how scientists think about the constraints and possibilities of biological complexity. A bat navigating a dark cave at full speed, among thousands of others, processes more real-time spatial data per second than most modern computers. Researchers have attempted to model the computational load of bat echolocation in dense environments, and the figures resist accommodation within any conventional framework of neural processing. The bat's brain, weighing under 2 grams, runs a sophistication that teams of engineers have spent careers attempting to approximate in silicon. It runs continuously for hours every night, while the bat is also flying, hunting, and avoiding predators. The lesson is not that bats are impressive. It is that brains shaped by 50 million years of selection solve problems in ways that make our best technology look like a rough draft. The bat is not the approximation. We are. A bat's wing is not a bird's wing. It is a human hand stretched thin, with skin webbed between elongated fingers. Hold your hand up. Spread your fingers wide. Now imagine the bones of your index, middle ring, and little finger growing to 30 centimeters in length, the skin between them stretching into a continuous elastic membrane, and your entire arm rotating at the shoulder with a range of motion no human joint could achieve. That is a bat wing. The scientific name for the wing membrane, the patagium, comes from a Latin word for a decorative border on a garment. The name is almost insultingly modest for what the structure actually is. Every fold, every curve, every tension in that membrane is controlled by the elongated finger bones beneath it, giving the bat a degree of wing-shape manipulation that no bird and no aircraft can match. A bat mid-flight is not flapping. It is sculpting the air, reshaping its wing geometry with every beat, every turn, every dive, using a modified version of the same hand you have. The wing membrane contains elastic fibers that store energy on the downstroke and release it on the upstroke like a biological spring. Flight is expensive. For an animal the size of a bat, the metabolic cost of sustained flapping could be prohibitive unless the wing itself helps pay the bill. The patagium is not simply a passive surface, it is an active energy storage system. As the wing moves downward on the power stroke, elastic proteins within the membrane, similar in structure to the elastin in human arteries, stretch and store mechanical energy. On the upstroke, that energy is released, helping to lift the wing back to position without requiring the full muscular effort the return stroke would otherwise demand. Engineers call this elastic energy return. Bats evolved it independently of birds, and their implementation is arguably more sophisticated because the membrane also changes shape between strokes, meaning the stored energy is released in a directionally useful way, not simply as rebound. The membrane does not simply flap. It participates. Some bats fly at over 160 kilometers per hour, the fastest horizontal flight speed recorded for any animal on Earth. For decades, the peregrine falcon held the popular title of fastest animal in flight. But the falcon achieves its record speed in a dive assisted by gravity wings partially folded, essentially falling with style. Horizontal sustained powered flight is a different challenge entirely. In that category, the Brazilian free-tailed bat, Tadarida brasiliensis, has been recorded at speeds exceeding 160 kilometers per hour in level flight under its own power, in a straight line, not diving. The bat achieves this through a combination of narrow, swept-back wings, optimized for speed rather than maneuverability, an exceptionally powerful flight muscle relative to body mass, and a body profile so aerodynamically refined that researchers initially questioned the tracking data when they first saw it. The fastest animal in horizontal flight weighs less than a standard envelope. The Mexican free-tailed bat migrates over 1,600 kilometers, navigating without GPS, without landmarks, using the Earth's magnetic field. Every autumn, populations of Mexican free-tailed bats in the southwestern United States travel south into Mexico, a journey covering the distance from New York to Miami, made at night over terrain that offers no reliable visual landmarks. By animals that weigh roughly 15 grams, how they navigate has been a subject of serious scientific investigation. The evidence points to magnetoreception, the ability to sense the Earth's magnetic field and use it as a directional reference. Bats have been shown to orient correctly in artificially manipulated magnetic fields in laboratory settings, losing that orientation when the field is disrupted. They likely also use star patterns on clear nights and low-frequency sound cues from geographic features. The journey is made annually by millions of animals, with navigational precision that matches purpose-built instruments, and it fits inside a skull the size of a walnut. Bats have a magnetic mineral magnetite embedded in their brain tissue, a biological compass that scientists are still mapping. Magnetite is an iron oxide mineral with permanent magnetic properties. It is found in the cells of migratory birds, in the lateral line of fish, in the bodies of bees, and it is present in bat brain tissue, specifically in regions associated with spatial processing and navigation. The magnetite crystals align with external magnetic fields, and the mechanical force of that alignment is thought to be transduced into a neural signal, giving the bat a continuous readout of magnetic field direction and potentially intensity. What scientists are still working out is precisely where in the brain this signal is processed, how it integrates with the bat's other navigational inputs, and what resolution the system achieves. Some researchers believe bats can detect not just the direction but the inclination of the magnetic field, which would give them latitudinal information as well. A compass and a rough map. Built from iron, running without power, without calibration, without error. During hibernation, a bat's heart rate drops from over 1,000 beats per minute to fewer than 20. At peak activity, a small bat's heart beats with a rapidity that makes a hummingbird seem measured over 1,000 times per minute in some species during flight. Then winter comes, insects disappear, and the bat enters torpor. Body temperature drops to within a degree or two of ambient cave temperature, sometimes close to freezing. Breathing slows to a few breaths per minute. Heart rate collapses to fewer than 20 beats. Metabolic rate falls to roughly 1% of active levels. The bat is not quite dead, but it is not in any meaningful sense fully alive. It exists in a suspended state, conserving every calorie, barely burning through its fat reserves, waiting. This transition is not gradual, it happens within hours, and the return from torpor full arousal to flight-ready status takes less than an hour, a rewarming process powered by specialized brown adipose tissue that generates heat without muscular shivering. A hibernating bat survives 6 months on stored fat, burning fewer calories per day than a single grape contains. The energy economy of bat hibernation borders on the miraculous. A small bat entering hibernation in autumn carries fat reserves amounting to roughly a third of its total body weight. Over the following 6 months, it will burn through those reserves at a rate so slow that the daily caloric expenditure is smaller than the energy content of a single grape. To sustain this, every physiological process that can be suspended is. Digestion stops. Immune activity drops to minimal maintenance levels. Even cellular repair processes slow dramatically. The risk is real. A bat disturbed during hibernation and forced to arouse early burns in minutes. The energy it would otherwise spend in weeks, and may not survive the winter if it cannot return to torpor quickly. White nose syndrome is so devastating partly because the fungus that causes it irritates hibernating bats into repeated early arousal, spending their energy reserves before spring, starving before insects return. A Brandt's bat was recorded at over 41 years old, a mammal the size of a thumb outliving most dogs. Body size and lifespan are strongly correlated in mammals. Larger animals live longer. A mouse lives 2 years, a rat 3 or 4. A bat weighing 6 grams, comparable to a large moth, should by the standard model live perhaps 4 or 5 years at most. Brandt's bat does not comply with this model. One wild individual recaptured in Russia decades after its original banding was confirmed at over 41 years of age. Other bat species show similarly extraordinary longevity, many living 20-30 years in the wild. For researchers studying the biology of aging bats represent a profound anomaly. Here is an animal that by every metabolic rule we understand should age rapidly: high heart rate, high body temperature during activity, high oxidative stress from flight, and yet it ages at a fraction of the rate its size predicts. Something in the bat's cellular biology has found a way around the standard cost of being alive. Scientists believe bats age slowly because of unique DNA repair mechanisms, research now informing human longevity studies. The leading hypothesis for bat longevity centers on unusually efficient DNA repair. During flight, bat cells experience elevated oxidative stress free radicals generated by intense metabolic activity, damage cellular machinery, including DNA. In most mammals, accumulated DNA damage is a primary driver of aging. Bats appear to have evolved repair enzymes of exceptional efficiency, catching and correcting oxidative damage before it accumulates to the threshold that drives cellular senescence. Comparative genomic studies have identified specific genes in long-lived bat species that show signatures of accelerated evolution genes involved in DNA damage response, telomere maintenance, and inflammation regulation. Several pharmaceutical research programs are now explicitly studying these bat-specific variants, looking for molecular insights that might translate into interventions for human aging. The bat has been solving the problem of growing old for 40 million years. We have only recently started paying attention. Bat immune systems run permanently in a state of controlled inflammation, neutralizing viruses before they can replicate. Most mammalian immune systems operate in two modes: resting baseline and activated response. The activation is triggered by infection, and it is expensive: fever, inflammation, metabolic cost. Bats appear to operate differently. Their innate immune system, the first-line non-specific defense, runs at a permanently elevated but tightly regulated inflammatory state. Interferons, the signaling proteins that alert cells to viral presence, are constitutively expressed in bat tissue at levels that would indicate active infection in most other mammals. This constant low-level readiness means that when a virus enters bat tissue, it encounters an immune environment already primed to suppress replication before the virus can establish a foothold. The virus persists, but at very low levels, unable to cause disease. The bat becomes a reservoir without becoming a victim. It is an immune strategy with no human equivalent, and we do not yet know how to build one. The same genes that power bat flight also regulate their immune response speed and disease resistance evolved together. This was one of the more unexpected findings to emerge from comparative bat genomics. When researchers sequenced the genomes of multiple bat species and looked for genes under accelerated evolutionary pressure genes that had changed faster than the neutral background rate, suggesting strong selection, they found the same gene. Families appearing in two contexts simultaneously. Genes involved in DNA damage response from the oxidative stress of flight, and genes involved in innate immune signaling. The overlap was not coincidental. The molecular machinery that bats evolved to manage the cellular damage of powered flight, the free radicals, the metabolic heat, the mechanical stress on wing tissue turns out to be intimately connected to the machinery that suppresses viral replication. Flight made bats healthier, or more precisely, the evolutionary pressure of flight forced a cellular housekeeping system so efficient that it incidentally became one of the most effective antiviral defenses in the mammalian world. The bat did not choose to be a reservoir host. It was an unintended consequence of learning to fly. A single little brown bat can consume over 1,000 mosquitoes in a single hour of hunting. 1,000! In 60 minutes. The little brown bat, Myotis lucifugus, weighs roughly 8 grams, fits comfortably in a closed-hand wingspan, about 25 centimeters. And yet, its hunting efficiency is extraordinary. Each insect pursuit takes a fraction of a second. The bat detects, closes, captures, swallows, and reorients in a continuous fluid sequence that researchers describe as almost mechanical in its economy of motion. On a productive night, a single bat may consume its own body weight in insects. Across a colony of even modest size, say 100 individuals, that is 100,000 mosquitoes removed from the local environment per hour. Across a summer season, the pest suppression delivered by a single small colony has been calculated to be worth hundreds of dollars in avoided agricultural pesticide costs. The bat charges nothing. Bats have a unique circulatory adaptation in their wings. Blood flow can be redirected mid-flight to regulate body temperature. The wing membrane presents a thermal challenge. It is a large, thin surface, excellent for heat dissipation, which is exactly what a bat needs during intense flight, but dangerous during cold nights when heat loss could become fatal. Bats manage this through a vascular system of unusual sophistication. The arteries and veins supplying the wing membrane run in close proximity, a countercurrent heat exchange arrangement that allows warm arterial blood to transfer heat to cooler returning venous blood before that blood reaches the wing to surface. When the bat needs to dump heat, blood flow to peripheral wing vessels increases. When it needs to conserve heat, flow is redirected away from the surface. This thermoregulatory valve is controlled autonomically. The bat does not consciously manage it, and it operates continuously throughout flight. The wing is simultaneously a propulsion surface, a sensory array, and a radiator. It is doing 3 jobs at once and doing all 3 well. Some bats enter daily torpor, a miniature hibernation lasting hours, to survive a single cold morning without food. Hibernation is the famous version. But many bat species practice a daily shorter version, entering torpor during daylight hours when temperatures drop and insects are unavailable, then rousing in the evening to hunt. This daily torpor is not simply sleep. Body temperature drops significantly, sometimes to within a few degrees of ambient air temperature. Heart rate and breathing slow. Metabolic rate falls to a fraction of resting levels. And then, as evening approaches and temperatures rise, the bat rewarms again, using brown adipose tissue, and emerges ready to fly within minutes. This flexibility, the ability to toggle between high-performance flight and near-metabolic suspension within the same day, represents a physiological range that no other mammal approaches. A bat, across 24 hours, may operate at both the highest and lowest metabolic states of any mammal. Its size, 2 speeds, 1 animal. Female bats can delay fertilization after mating, storing sperm for months until conditions are right for pregnancy. In temperate bat species, mating often occurs in autumn before hibernation. But pregnancy during winter is impossible. The metabolic demands of gestation cannot be met when the bat is in torpor and insects are unavailable. The solution is remarkable. After mating, the female stores viable sperm in her reproductive tract, sometimes for up to 200 days, maintaining it in a metabolically quiescent state through the winter. When she emerges in spring and insects become available, fertilization occurs timed precisely to align gestation and birth with peak insect abundance. This sperm storage is not passive. The female's reproductive tract actively maintains sperm viability, nourishing the cells, preventing premature fertilization, and regulating the timing of ovulation with apparent environmental sensitivity. What is clear is that the female bat is not simply waiting, she is managing a reproductive process across months, with a precision that defies the simplicity we tend to attribute to small mammals. Bat pups are born knowing nothing, yet within weeks they learn echolocation by listening to their mothers, a form of vocal learning shared with humans and songbirds. A newborn bat pup cannot echolocate. It can produce simple contact calls, high, thin sounds that alert the mother to its location. But the sophisticated frequency-modulated pulses of adult echolocation must be learned. Researchers have documented a process strikingly parallel to human language acquisition. The pup begins with acoustic babbling producing variable, exploratory vocalizations that gradually narrow toward the adult call structure through a combination of self-monitoring and, crucially, exposure to the mother's calls. Species-specific call structure is not hardwired. It is learned. And like human children acquiring language, bat pups show a sensitive period, a developmental window, during which acoustic input is most effectively incorporated. Miss that window through isolation or deprivation, and the adult call structure is compromised. Vocal learning, the ability to modify vocalizations based on auditory feedback, was long considered a rare capacity. Bats, humans, and songbirds share it. The company is worth considering. The bones inside a bat wing are hollow, thin as paper, yet flexible enough to survive the mechanical stress of 50,000 wing beats per night. Bone fatigue is well understood in engineering. Any material subjected to repeated cyclical stress develops microscopic cracks that propagate and ultimately cause failure. For bat wing bones, the numbers are daunting. A bat in active flight beats its wings 10 to 17 times per second. That accumulates to tens of thousands of stress cycles on bones, measured in fractions of a millimeter thickness. And yet bat bones do not fail under normal conditions. The explanation lies in their microstructure, an unusually high proportion of cortical bone with mineral composition and crystal orientation optimized to resist the specific bending forces of the wing beat cycle. They are not simply thin versions of normal bones. They are bones redesigned at the crystalline level for a single mechanical task. Bats lose body heat through their wing membranes, and the same surface that cools them in summer helps them read the air they fly through. During intense flight, a bat's core body temperature rises sharply. The metabolic heat generated by continuous muscle activity must be shed faster than a small furry body can manage through conventional means. The wing membrane solves this richly vascularized, thin enough for heat to transfer rapidly to surrounding air, large relative to body size. On cold nights, the countercurrent vascular system recaptures heat before it reaches the surface. On warm nights, the membrane flushes with blood, and the bat flies inside its own cooling system. But the membrane is doing something else simultaneously. Its embedded sensory cells are reading pressure gradients, airflow shifts, the subtle aerodynamic signatures of turbulence and stall, cooling the body and sensing the sky. Through the same skin. At the same moment, scientists still aren't sure how bats sleep upside down without falling. The tendons lock passively, but the neurological trigger remains poorly understood. The mechanical explanation is known. Bats have a tendon arrangement in their feet such that the weight of the body pulling downward causes the tendons to tighten around the toe bones, creating a grip that requires no muscular effort to maintain. The bat can be fully unconscious and still hang securely. Release requires active muscular contraction, meaning a dead bat, paradoxically, does not fall. But the deeper question, how the bat manages the transition from aroused flight to suspended torpor, without losing its grip at the moment of muscular relaxation, involves a neural coordination sequence that has not been fully characterized. There is also the matter of blood pressure. Hanging inverted with the brain below the heart should create cardiovascular challenges. Bats appear to have vascular adaptations that manage this. An animal this thoroughly studied still holds genuine physiological mysteries. Some questions, the more you look, only get larger. The tube-lipped nectar bat of Ecuador has a tongue longer than its entire body coiled inside its chest cavity. Take a moment with that. Not longer than its head, longer than its entire body. Anora fistulata, a bat that by any reasonable anatomical standard should not be possible. The tongue, when fully extended, reaches 1.5 times the bat's body length. There is not enough room in the mouth to store it, so it retracts into the chest cavity instead, coiling between the heart and sternum. Anchored by a modified hyoid bone structure that extends further into the thorax than in any other bat species. The tongue evolved to reach the nectar at the base of Centropogon nigricans, a flower whose corolla is so deep that no other pollinator on Earth can access it. The bat and the flower shaped each other across millions of years, the flower growing longer, the tongue following until the relationship became exclusive. Neither can fully reproduce without the other. It is dependence made biological, a partnership written in anatomy sealed over millennia and still ongoing every night in the Ecuadorian cloud forest. A bat's thumb, the small hook at the top of the wing, is the only digit not incorporated into the membrane, and bats use it for climbing, gripping, and manipulating their world. Look closely at a bat wing, and you will find it tucked at the leading edge, a small curved claw free of the membrane, independently mobile. In the developmental sequence that produces the bat wing, 4 of the 5 digits elongate dramatically and become incorporated into the patagium. The thumb alone is exempted. It remains short, clawed, and functional, a remnant of the ancestral mammalian hand that never got folded into the flight apparatus. Bats use it constantly, to shuffle along cave ceilings, to grip prey items briefly before swallowing, to manipulate roosting positions. In vampire bats, the thumb claw is muscular and weight-bearing, used to walk, run, and even hop on the ground with surprising agility. A locomotion style, almost unique among bats. In species that roost in foliage, it hooks into bark or leaf stems. It is a small structure, easily overlooked. It is also the last surviving piece of the hand that flight did not consume. Some bats can fly in near freezing temperatures, keeping wing muscles warm through a specialized fat metabolism that operates differently from any other mammal. Most muscles lose efficiency as temperature drops the biochemical reactions driving contraction slow. The mechanical properties of the tissue stiffen and performance degrades. For a bat hunting on a cold night, this is an existential problem. Wing muscles that cannot contract rapidly enough cannot generate sufficient lift. The solution bats evolved involves brown adipose tissue, not just as a heat reservoir for arousal from torpor, but as an active thermogenic system that pre-warms the thoracic muscles before and during flight. In some species, the flight muscles are anatomically surrounded by brown fat deposits that generate heat on demand, maintaining muscle temperature independently of ambient conditions. The lipid composition of bat muscle cell membranes also differs from other mammals, with a higher proportion of unsaturated fatty acids that maintain membrane fluidity at lower temperatures. The bat, in effect, carries its own heating system wrapped around its engine. Cold nights are an inconvenience, not a barrier. The skin of a batwing is among the thinnest biological membranes ever measured, yet heals from tears within 3 weeks. The patagium is approximately 0.1 millimeters thick, in many species thinner than a sheet of standard printer paper, thinner than most food wrap. Under a microscope, it is a remarkable structure, a sandwich of 2 skin layers enclosing a network of blood vessels, sensory neurons, elastic fibers, and muscle cells, all compressed into a membrane that must survive the mechanical punishment of thousands of wing beats nightly. Tears happen. Thorns rough roost surfaces, the claws of other bats, the membrane is vulnerable, and yet repair occurs with a speed and completeness that has surprised researchers studying wound healing. The patagium regenerates across gaps that would leave permanent scarring in human skin, restoring both structural integrity and the sensory nerve network within weeks. The mechanism involves a high density of stem cell-like progenitor cells distributed throughout the membrane, a regenerative capacity that is now being studied in the context of human wound healing research. What the bat does routinely, we have only recently learned to want. Researchers studying bat flight believe the bat wing remains more aerodynamically efficient than any human-made equivalent at its scale. At small scales, wingspans under half a meter, flight speeds under 50 kilometers per hour, the physics of airflow create challenges that rigid wing designs handle poorly. Bat wings sidestep these challenges through continuous, active deformation. The membrane changes shape dynamically with every wing beat, not because the bat consciously adjusts each parameter, but because the elastic membrane and finger bone structure passively respond to aerodynamic forces in ways that maintain optimal airflow. Engineers building micro-air vehicles have repeatedly found that rigid designs hit efficiency ceilings that bat-inspired flexible membranes do not. Several research programs now build drone wings explicitly modeled on bat membrane microstructure. And still, in every meaningful performance metric at this scale, the biological original leads. The engineers are not catching up. They are taking notes. There are over 1,400 known species of bats, making up roughly 20% of all mammal species on Earth. Step back from the individual bat for a moment and consider the group. 1,400 species. There are roughly 300 species of primates, fewer than 300 species of carnivores. Bats have diversified into an extraordinary range of ecological roles, body plans, dietary strategies, and geographic ranges. They occupy every continent except Antarctica. They live in deserts, rainforests, caves, bridges, tree hollows, and the foliage of urban parks. They range in size from the bumblebee bat, the world's smallest mammal, by mass weighing under 2 grams, to flying foxes, with wingspans approaching 2 meters. They eat insects, fruit, nectar, fish, frogs, other bats, and blood. This diversity reflects 50 million years of a single body plan, radiating into every available niche, adapting to every available food source, solving every available ecological problem with the tools evolution provided. The bat is not a creature, it is a category. A single colony of 30 million Mexican free-tailed bats eats over 200 tons of insects per night and saves billions in crop losses without sending a single invoice. 200 tons per night. The Bracken Cave colony in Texas has been the subject of detailed feeding studies, and the numbers that emerge resist easy comprehension. Each bat consumes roughly its own body weight in insects nightly. The colony aggregated removes more insect biomass from the surrounding landscape in a single evening than most agricultural pest control programs achieve in a season. A peer-reviewed economic analysis estimated that insectivorous bats provide over $22 billion annually in avoided crop losses and reduced pesticide costs in North America alone, a figure that does not include the downstream costs of increased pesticide use, water contamination, or soil health degradation. Expanded globally, the estimate reaches into the hundreds of billions. These are not hypothetical numbers. They are what happens when a service that has always been provided for free is priced at the cost of replacing it. The bat does not invoice anyone. The accounting only happens when the bat is gone. The insects bats prefer to hunt are not random. They target agricultural pest moths, whose larvae cause billions in crop damage annually. This is the detail that transforms the economic argument from impressive to urgent. Mexican free-tailed bats do not hunt indiscriminately. Studies of gut content and prey selectivity show a strong preference for soft-bodied moths, and among those moths, a disproportionate representation of species whose larvae are primary agricultural pests. The corn earworm moth, the cotton bollworm, the tobacco budworm. These are not peripheral species. They are among the most economically destructive insects in North American agriculture, responsible for crop losses that run into billions annually, even with current pesticide regimes. The bats are not simply eating whatever is available. They are night after night, targeting the specific insects that farmers spend the most money trying to suppress. Whether this preference is driven by abundance, palatability, or flight pattern is still being studied. What is not in question is the outcome. The bat hunts the pest. The pest damages the crop. Remove the bat, and the math changes fast. There are exactly 3 species of vampire bat, and all 3 live only in Latin America. Among 1,400 species, 3 are sanguivores: Desmodus rotundus, the common vampire bat; Diemus youngi, the white-winged vampire bat; Diphila ecordata, the hairy-legged vampire bat. Each specializes. The common vampire bat feeds primarily on mammal blood, while the white-winged and hairy-legged species prefer birds. All 3 are found only in the Americas. From Mexico to Argentina, the evolutionary origin of blood feeding in bats is debated. Some researchers believe it derived from an insectivorous ancestor that began feeding on open wounds of large animals, gradually specializing as the behavior proved reliably profitable. Blood is nutritionally a protein and lipid-rich liquid food, not entirely unlike the soft-bodied insects that most bats consume. The delivery mechanism changed. The basic dietary equation did not. Vampire bats don't suck blood, they make a small incision and lap, while an anticoagulant in their saliva keeps the wound open for up to 30 minutes. The folklore is wrong about the mechanism. Vampire bats have razor-sharp upper incisors that can remove a small disc of skin with a precision the host animal, if large enough, often does not wake to feel. There is no suction. The bat laps the pooling blood with a grooved tongue that channels flow efficiently into the mouth, while saliva containing the anticoagulant compound draculin continuously prevents clotting at the wound site. Draculin inhibits the coagulation factors responsible for clot formation, keeping blood fluid and freely flowing for far longer than the wound would naturally remain open. The bat feeds for approximately 30 minutes before departing. The host loses a modest amount of blood. The wound closes slowly. The animal may never know it happened. Vampire bat saliva contains a compound now being studied as a stroke treatment because of its precision in dissolving blood clots without triggering wider bleeding. Draculin has attracted serious pharmaceutical interest. Stroke caused by arterial blood clots is one of the leading causes of death and disability worldwide, and the primary treatment tissue plasminogen activator has a narrow therapeutic window and significant bleeding risk. Draculin works differently. Rather than broadly activating the clot-dissolving cascade, it inhibits specific coagulation factors with a selectivity that leaves the rest of the clotting system largely intact, potentially offering clot dissolution with lower bleeding risk. A modified version of the compound renamed desmoteplase after the bat's Latin name entered clinical trials for ischemic stroke treatment. Results have been mixed, and the research is ongoing. A bat that evolved to keep blood flowing in its prey may yet help keep blood flowing in human brains. The molecular logic holds. Whether the medicine follows is still being worked out. Vampire bats share blood meals with colony members who failed to feed, a form of reciprocal altruism observed in very few non-human species. A vampire bat that fails to feed on a given night faces a genuine survival crisis. Unlike most bats, which can draw on fat reserves during lean periods, vampire bats have a very limited metabolic tolerance for fasting. They can survive roughly 60 hours without blood before the consequences become fatal. The colony's response to this vulnerability is extraordinary. Bats that fed successfully will regurgitate blood meals to share with hungry roost mates, including individuals who are not close relatives. This is not simple kin-based generosity. Studies by researcher Gerald Wilkinson demonstrated that sharing occurs preferentially between individuals with a history of mutual exchange. Bats that have shared with a partner in the past are more likely to receive sharing in return. The colony maintains in effect a social ledger. Favors are recorded and reciprocated. Cheaters bats that accept meals but never reciprocate are gradually excluded from the exchange network. Reciprocal altruism of this sophistication, operating across non-kin relationships, has been documented in humans, primates, and vampire bats. The common vampire bat can recognize individual humans by their breathing patterns and return to the same host night after night. Individual recognition from breathing alone requires distinguishing the subtle acoustic signatures that differentiate one person's respiratory pattern from another, variations in rate, depth, rhythm, and the faint sounds of air moving through a specific nasal and throat geometry. Vampire bats have been documented doing exactly this. In studies of bat predation on human sleepers in rural Latin American communities, individual bats showed strong preferences for specific people within a household, returning repeatedly to the same host while ignoring others sleeping nearby. The preference appears stable across nights, suggesting genuine individual recognition rather than opportunistic selection of the easiest target. The bat memorizes you, not your face, not your smell, though that likely contributes. Your breath, the specific acoustic signature of your sleeping respiratory pattern distinguishable from every other person in the room. There is something deeply unsettling about being known that intimately by something you cannot see, have never seen, and have no awareness of at all. Fruit bats carry seeds in their gut and deposit them kilometers from the parent tree. Some tropical tree species depend entirely on bats for seed dispersal. A fruit bat eating a fig in the forest canopy is not simply feeding, it is providing a transportation service. Seeds swallowed whole pass through the gut, largely intact, and are deposited in viable condition wrapped in a small parcel of fertilizer at whatever location the bat happens to be when digestion completes. For large seeded tropical fruits, the bat may be the only animal capable of swallowing the seed whole and carrying it far enough from the parent tree to give the seedling a realistic chance of establishment. In forests where large fruit-eating birds and mammals have been hunted out, bats often become the sole remaining seed dispersers for entire guilds of tree species. Remove the bats, and those trees stop recruiting new individuals. The canopy over decades slowly loses species. Seed dispersal is one of those services that registers only in its absence, by which point the trees that needed it are already gone. The baobab tree, one of the most iconic and ancient trees on Earth, relies almost exclusively on bats for pollination. The baobab opens its flowers at night, large white pendulous blooms that hang from branches like lanterns releasing their scent after dark, a scent described by botanists as fermenting or slightly sour, precisely calibrated to attract fruit bats rather than daytime pollinators. The flowers produce abundant nectar and large quantities of pollen, and the bat pushing its face into the bloom to feed emerges dusted in yellow. It carries that pollen to the next baobab it visits, cross-pollinating trees that may be separated by considerable distances in the sparse African savanna. Some baobabs alive today are estimated to be over 2,000 years old. They have been pollinated by bats for the entirety of that span, every generation of fruit, every seed, every new tree dependent on a nocturnal mammal completing a transaction in the dark that the tree cannot complete itself. The baobab is ancient and enormous and seemingly immovable, and it cannot reproduce without a creature that weighs less than a mango. Over 500 plant species require bat pollination to reproduce, including the agave plant, the source of tequila. 500 species is not a footnote. It is a significant portion of tropical and subtropical plant diversity, trees, shrubs, cacti, and flowering plants across multiple continents whose reproductive cycles are timed to bat activity, whose flower shapes are sized to bat faces, whose nectar chemistry is calibrated to bat preferences. The agave is perhaps the most economically visible example. Wild agave populations in Mexico depend on the lesser, long-nosed bat, Leptonicteris yerbabuena, for pollination. The bat's long snout and tongue fitting precisely into the agave flower's architecture. Commercial agave farming, which harvests plants before they flower to maximize sugar content for tequila production, has progressively reduced the wild pollinated agave population and the bat population that depends on it. A feedback loop that threatens both the bat and the genetic diversity of the agave itself. Researchers studying this decline use a phrase worth sitting with: ecological debt. The cost accumulates invisibly, paid in species lost in genetic diversity, reduced in resilience, quietly eroded, until the system can no longer absorb the next disruption. The giant golden-crowned flying fox of the Philippines has a wingspan of 1.7 meters, roughly the width of a standard doorway. Acerrodon jubatus. It does not echolocate. It navigates by sight and smell, its large eyes adapted for low-light conditions, its olfactory system tuned to the scent of ripe fruit from considerable distances. It roosts in colonies in old-growth forest, hanging from large trees in clusters that can number in the thousands. A social, visible, audible presence in the forest canopy. And it is critically endangered. The primary threats are direct hunting for bushmeat habitat destruction as old-growth forest is cleared and disturbance of roost sites. The species has no meaningful legal protection. Across much of its range and its slow reproductive rate, females produce a single pup per year, means that populations recover from hunting pressure with agonizing slowness, if at all. A bat with a wingspan nearly as wide as you are tall, which has been roosting in Philippine forests for millions of years, may disappear within decades, not because of any complexity or subtlety in the threat, but simply because it is large, enough to be worth shooting, and nobody stopped it. Flying foxes eat only fruit, flowers, and nectar, and humans have hunted them toward extinction across much of their range. The flying fox is the least threatening large bat imaginable. It does not carry rabies in most of its range. It does not bite unprovoked. It does not prey on livestock or crops. In net negative terms, the seed dispersal and pollination services it provides to the forest ecosystem far outweigh any fruit consumption from cultivated trees. And yet, it is hunted. In Guam, Palau, and parts of Indonesia, flying foxes have been consumed as a prestige food for generations, and colonial-era introductions of firearms dramatically increased kill rates beyond sustainable levels. In Guam, 2 endemic flying fox species were hunted to extinction within decades of firearms becoming widespread. Several more species across the Pacific are now functionally extinct in the wild. What disappears with them is not just the animal, it is the forest regeneration service, the pollination network, the ecological function that no other animal in those islands performs. The tree that loses its flying fox loses something it cannot replace. Some bats hunt fish, detecting a fin breaking the water surface from 3 meters above in complete darkness. The greater fishing bat, Noctilio leporinus, has feet like grappling hooks, elongated curved claws on powerful hind legs trailing below the body during low passes over water. It flies just above the surface echolocation, scanning the interface between air and water, reading the tiny acoustic anomalies that a protruding fin or a fish moving just below the surface creates in the echo signature. The detection threshold is extraordinary. A fin breaking the surface by just a millimeter or two produces a detectable signal at several meters distance. The bat then times its foot deployment with precision, raking the water at the exact coordinates the echo indicated, at the exact moment of closest approach, a ballistic problem solved in real time, in darkness at flight speed. Successful catches are swallowed in flight or carried to a perch. Failed passes are abandoned, and the bat reorients immediately. The entire sequence detection approach strike evaluation takes under 2 seconds. Fishing for this bat is not patience, it is geometry. The spectral bat of Central America is the largest carnivorous bat. It hunts other bats in flight, using silence and surprise. Vampirum spectrum. Despite the name, it is not a vampire bat. It does not feed on blood. It is a predator of birds, rodents, and other bats, with a wingspan reaching 90 centimeters and a bite force sufficient to crack small bird skulls. What makes it remarkable as a hunter is its acoustic stealth. The spectral bat has largely reduced its echolocation during hunting, emitting calls of very low intensity that minimize its acoustic signature, making it difficult for prey bats to detect using their own sonar detection systems. It hunts in the forest understory, moving slowly and quietly until close enough to strike. Researchers who have studied spectral bat predation describe prey bats that showed no evasive response until physical contact, suggesting the predator approached within striking range without triggering the prey's threat detection. In a world where sound is the primary sensory currency, silence is the most lethal weapon available. The spectral bat found this and built a hunting strategy around it. In some cave ecosystems, bat guano accumulated over centuries becomes the nutritional foundation for an entire independent food web. Guano, the accumulated feces of thousands or millions of bats, builds up on cave floors over decades and centuries into deep, nutrient-rich deposits. In caves where no photosynthesis occurs and no plant life penetrates, guano is the only significant energy input into the ecosystem. And it supports one. Specialized beetles, flies, mites, and springtails colonize guano deposits, feeding directly on the organic material and the bacteria and fungi that decompose it. These invertebrates are preyed upon by cave spiders, cave-adapted centipedes, and specialized cave fish in subterranean streams fed by guano-enriched water. In Gomantong Cave in Borneo, the guano ecosystem supports millions of invertebrates and has been studied as one of the most complete examples of a food web running entirely on biological input rather than solar energy. Remove the bats from above, and the entire ecosystem below collapses within a season. The cave is not a backdrop. The bat is the sun it runs on. Bats have natural predators: owls, hawks, snakes, and in some cave systems, large centipedes that hang from ceilings and snatch them mid-roost. Owls are the primary aerial threat, silent flyers themselves operating in the same darkness, hunting by sound with an auditory system nearly as sophisticated as the bat's own. Hawks take bats at emergence, the brief vulnerable window when millions of animals funnel out of a cave entrance in a dense column, creating an opportunity that raptors exploit with precision. Snakes position themselves at roost entrances, striking at bats as they exit or return. And then there are the centipedes. In caves in Venezuela and Trinidad Scolopendra gigantea, a centipede reaching 30 centimeters in length, has been documented hanging from cave ceilings, by its rear legs dangling into the flight path of exiting bats, seizing them with its front legs and delivering a venomous bite. This behavior, documented on camera for the first time in 2005, required a fundamental reassessment of what centipedes are capable of as predators. The bat, for all its sophistication, exists within a web of threats from which no amount of sonar provides complete immunity. Scientists still debate the full extent of bats' role in insect population regulation. Some models suggest they prevent explosions that would otherwise devastate temperate agriculture. The direct feeding studies are clear enough: individual bats consume specific quantities of specific insects and colonies, aggregate those numbers into ecologically significant totals. What remains genuinely contested is the landscape-level effect. Does bat predation actually suppress insect populations below the thresholds at which outbreaks occur? Or do bats simply harvest insects at rates that track population size without driving it down? The answer matters enormously for conservation policy. If bats are merely taking insects that would otherwise die of other causes, a kind of harvesting at the margin, then their loss, while regrettable, may not cascade into agricultural crisis. If they are actively suppressing populations that would otherwise erupt, then their loss removes a regulatory function that no other predator replicates at equivalent scale. The evidence increasingly favors the second interpretation, particularly for moth species, whose larvae are primary agricultural pests. But the models are complex, the field data patchy, and the full picture remains genuinely open. In a world without bats, the ecological cascade would reach dinner plates within a decade. This is not hyperbole. It is a summary of what the models produce when you remove bat-delivered pest suppression from agricultural systems in temperate North America, Europe, and Asia simultaneously. Corn earworm, cotton bollworm, soybean looper, moth species, whose larvae are primary drivers of crop loss, would face reduced nocturnal predation pressure. Populations would rise, crop damage would increase. Pesticide application rates would follow. The insects would develop resistance to those pesticides, as they always do, faster than alternatives can be developed. Yields would fall, food prices would rise. The timeline to measurable consumer impact in crop availability and grocery cost has been modeled at under a decade from the point of significant bat population collapse. The bat is not a luxury of the natural world, it is structural. It is load-bearing. Bracken Cave in Texas houses the largest bat colony on Earth, 15 million Mexican free-tailed bats, the most densely packed mammal aggregation anywhere on the planet. The number alone is inadequate preparation for the reality. 15 million individual mammals occupying a single cave system in the Texas Hill Country, representing a biomass concentration with no equivalent in the mammalian world. The cave itself is not large by geological standards, a modest entrance, a descending chamber, passages that branch into roost areas, packed so densely that observers describe the ceiling as appearing to move as a continuous rippling surface of fur and membrane. The smell reaches visitors before the entrance does. The sound a continuous layered roar of millions of individual calls, wing beats, and social vocalizations is described by researchers who have entered the cave as physically disorienting. It is not a place designed to accommodate human perception. It is calibrated for something entirely different. 15 million animals operating in a sensory register we cannot access in a social density, we cannot imagine in a darkness we cannot navigate. It is theirs. We are merely visitors who learn to write down what we found. When the Bracken Cave colony emerges at dusk, the flight column is visible on weather radar and has been mistaken for storm systems by meteorologists. The emergence takes approximately 3 hours. From a single cave entrance, 15 million bats pour into the evening sky in a continuous column that spirals upward and disperses across the surrounding landscape in a fan that can extend over 100 kilometers. The biomass in motion, the sheer physical volume of animals, creates a radar return indistinguishable at first glance from a developing storm cell. National Weather Service radar archives contain multiple instances of the Bracken Cave emergence, appearing as an anomalous radar signature, diffuse organic in shape, expanding outward from a fixed point in a pattern that doesn't match precipitation or wind patterns. Meteorologists in the region now recognize it. But the fact that it registers at all, that the flight of living animals can produce a signal detectable by instruments designed to track water vapor, gives some indication of the physical scale of what happens at that cave entrance every evening between March and October. Biology at sufficient concentration becomes weather. Inside Bracken Cave, bat pup density reaches over 4,000 individuals per square meter, the most densely packed mammal nursery ever documented. The maternity colony forms in late spring when pregnant females return from their wintering grounds in Mexico. They give birth in the cave synchronously within a window of a few weeks, and the pups accumulate on the ceiling in creche, clusters of extraordinary density. 4,000 pups per square meter, each one approximately the size of a large grape, hairless at birth, clinging to the rock surface with feet that have been gripping since the moment of birth. They cannot fly. They cannot echolocate effectively. They are entirely dependent, and they are surrounded by 4,000 others of their kind per square meter in complete darkness in a cave that smells of ammonia and guano, generating collective body heat that raises the local temperature significantly above ambient. It is simultaneously one of the most extreme nursery environments on Earth, and by mortality metrics, one of the most successful. Pup survival rates in well-established maternity colonies are remarkably high, a testament to the thermal benefits of density and the precision of maternal recognition. A mother bat returning to a creche of millions can locate her own pup using echolocation scent and a vocal signature the pup learned within hours of birth. The problem she faces would defeat most search algorithms. She returns from hunting to a cave containing millions of pups clustered at densities that make individual identification seem impossible, and yet she finds hers with a reliability that approaches 100% under normal conditions. The mechanism is multi-layered. The pup begins producing an individual contact call within hours of birth, a vocalization with a frequency pattern and temporal structure specific enough to be distinguishable from thousands of other pups calling simultaneously. The mother learns that call. When she returns, she calls and listens for the response. She narrows her search using the call location, then confirms identity at close range using olfactory cues. The pup's individual scent, which she also memorized during the first hours of contact. Echolocation provides the final spatial precision to locate the exact position on the ceiling. 3 independent recognition systems operating in sequence, each providing a different resolution of the same answer. The mother does not guess. She knows. Bat pups produce babbling sounds before developing their adult calls, a vocal learning process structurally similar to human infant language acquisition. The parallel was identified formally by researchers studying the greater sac-winged bat, Sacopteryx bilineata, in Costa Rica and Panama. Pups produce long, rambling vocal sequences, repetitive variable exploratory, that contain fragments of adult call types mixed with novel combinations that appear nowhere in the adult repertoire. The sequences gradually narrow over weeks as the pup's vocalizations converge toward the species-specific adult pattern through a combination of self-monitoring and exposure to adult calls. The structural similarity to human-infant babbling is not superficial. Both involve the production of variable vocal output during a sensitive developmental period, gradual convergence toward a target structure through auditory feedback, and the emergence of individual vocal identity within a species-typical framework. The question worth sitting with is not that they converged. It is what that convergence says about the problem itself. Whether there are only so many ways to wire a brain for language, and bats and humans both found the same narrow door. Mother bats remember their pup's call for life, even after the pup has grown and no longer needs feeding. This was demonstrated in long-term studies of greater horseshoe bats, where individually marked females were observed to respond preferentially to the calls of their adult offspring years after weaning, orienting toward them in flight, roosting preferentially in their proximity and showing reduced alarm responses in their presence compared to unrelated individuals of equivalent age. The mother does not forget. The acoustic signature she memorized in the hours after birth remains in her recognition system for the remainder of her life. A memory that serves no obvious ongoing functional purpose once the pup is independent yet persists anyway. What this suggests about bat memory, about the depth and permanence of the social information stored in a brain weighing less than 2 grams, sits uncomfortably with simple models of animal cognition. The bat remembers her child, not as an abstraction, as a sound, a specific individual irreplaceable sound that she has carried in her neural architecture undiminished for years. Bat colonies thermoregulate collectively the body heat of millions of individuals, raising cave temperature enough for pups to survive without their mother's warmth. A pup left alone in a cold cave would lose body heat rapidly. It lacks the fat reserves and thermoregulatory capacity of an adult, and hypothermia would be fatal within hours. The creche solves this. Thousands of pups clustered together generate collective body heat that elevates the local microclimate significantly above ambient cave temperature in Bracken Cave. The maternity roost area can be measurably warmer than the surrounding passages purely from metabolic heat. This thermal benefit is one of the primary evolutionary drivers of colonial roosting. The larger the colony, the more stable and elevated the thermal environment, the higher the pup survival rate. It is thermoregulation without any individual effort, a benefit that emerges from density alone, requiring no coordination, no communication, no awareness. No individual bat is trying to heat the cave. The cave heats anyway. Some bat colonies roost in the same cave for over 100,000 years, the guano layers below serving as a geological record of their presence. 100,000 years. That is longer than anatomically modern humans have existed as a species. While our ancestors were still navigating the last ice age, still developing the cognitive architecture that would eventually produce language and art and agriculture, certain bat colonies were already established in their caves, already depositing the guano layers that researchers can now core like ice or sediment, reading the chemical and biological record compressed within them. Guano cores from caves in Europe and Asia have been dated to over 100,000 years before present, with continuous deposition suggesting unbroken colony occupation across that entire span. The record within those layers is remarkable pollen grains that document the vegetation surrounding the cave across millennia, insect chitin fragments that reflect the prey available in different climatic periods, isotopic signatures that track temperature and humidity through glacial cycles. The bat did not merely survive those changes, it recorded them. Every night's feeding compressed into centimeters of geological time, waiting for a researcher with a coring tube to come and read it. When a section of a colony is threatened, alarm signals propagate through the roost faster than any individual bat could physically travel a distributed early warning system. The mechanism is not fully understood, but the observation is consistent. A disturbance at one end of a large roost, a predator entering a researcher approaching an unfamiliar sound, produces a wave of agitation that moves through the colony faster than the disturbance itself propagates through the air. Bats at the far end of the roost begin shifting, vocalizing, and preparing for flight before any direct stimulus has reached them. The signal travels bat to bat, acoustic, visual, and possibly olfactory cues cascading outward from the point of disturbance in a chain reaction that covers hundreds of meters of roost in seconds. Each bat responds to its immediate neighbor's behavior rather than to the original threat, producing a collective alarm response that amplifies and spreads faster than any centralized communication could achieve. No individual bat knows what the others know. The alarm travels anyway. Bats groom one another, a social bonding behavior that removes parasites, reduces stress, and reinforces colony relationships in ways that parallel primate grooming. Allogrooming of one individual by another is widespread in social mammals, and its functions extend beyond hygiene. In primates, grooming is the primary currency of social bonding, alliance building, and tension reduction. Time spent grooming correlates with the strength of social relationships and with stress hormone levels in both groomer and recipient. Bats show the same pattern. Roostmates groom each other's fur, ears, and wing membranes, areas the individual cannot easily reach alone, and the behavior correlates with measures of social affiliation and colony stability. Individuals that receive more grooming show lower stress indicators. Grooming pairs tend to share roost positions, food information, and alarm responses preferentially. The social architecture of the bat colony, who trusts whom, who cooperates with whom, is partly written in grooming relationships. The bat is not simply maintaining its fur. It is maintaining its social world. Some bat species form long-term social bonds. Female greater horseshoe bats have been observed roosting with the same companions for over a decade. Long-term studies tracking individually marked greater horseshoe bats in the United Kingdom have documented roost-mate associations that persist for years, the same 2 or 3 females consistently choosing proximity within the roost across successive seasons despite the disruption of hibernation, migration, and annual reproductive cycles that could plausibly reset social preferences. A decade-long association in an animal with a lifespan of 20-plus years is proportionally equivalent to a multi-decade human friendship. What maintains these bonds across the seasonal disruptions is not fully understood. Olfactory recognition likely plays a role. Bats have individual scent profiles and can distinguish familiar from unfamiliar individuals by smell. Call recognition persists, as established by the maternal memory studies. Perhaps the bond is simply the accumulated residue of repeated positive interactions, the bat equivalent of a relationship built from years of small, unremarkable moments of proximity. Science does not yet have a language for what a decade-long bat friendship actually consists of, but the data says it exists. And that is enough to ask the question. Bats communicate with a repertoire of over 25 distinct call types, encoding identity, location, emotional state, and social rank. The echolocation calls are only part of the acoustic picture. Bats produce a parallel repertoire of social calls, lower frequency vocalizations, many within human hearing range, used to communicate within the roost and during flight. These calls have been analyzed spectrographically across multiple species, and the complexity revealed is considerable. Distinct call types are associated with specific social contexts: aggressive encounters, roosting aggregation, mother-pup contact, mating approaches, foraging information, sharing, and distress. Individual identity is encoded in the acoustic structure of calls. Bats can recognize familiar individuals by voice alone. In some species, call structure correlates with dominance status. Higher-ranking individuals produce calls with different frequency characteristics that subordinates appear to respond to differentially. Whether this constitutes a primitive form of social signaling analogous to vocal status indicators in primates is a question researchers are actively exploring. The bat is not simply making noise in the dark, it is speaking. The vocabulary is different, but the intent is not. Scientists have identified what may be bat dialects, regional variations in call structure between colonies of the same species passed down through generations. In human populations, dialects emerge when geographically separated groups develop slightly different versions of a shared language, driven by social isolation, local innovation, and the gradual drift of acoustic norms within a community. Researchers studying greater horseshoe bats across their European range have identified analogous patterns. Colonies separated by geography show subtle but consistent differences in call structure that cannot be explained by genetic differences alone, and that appear to be transmitted culturally, rather than encoded in DNA. Pups raised in isolation produce calls that differ from their natal colony's dialect, suggesting the dialect is learned, not inherited. Cross-fostering experiments where pups are raised by mothers from a different colony produce adults whose calls more closely resemble their foster colony's dialect than their genetic colonies. The calls are being learned. The regional variants are being transmitted. Whether this constitutes dialect in the linguistically meaningful sense is debated, but the mechanism, the transmission pattern, and the geographic distribution are all consistent with cultural inheritance of acoustic norms. The bat colony is not just a roost, it may be a culture. When white-nose syndrome struck North American bat populations, some colonies lost over 90% of members in a single winter. Pseudogymnoascus destructans, the fungus responsible, was introduced to North America from Europe, likely carried on the equipment or clothing of cave explorers sometime around 2006. European bats have co-evolved with the fungus for long enough to have developed partial resistance. North American bats had no evolutionary exposure and no resistance. The fungus colonizes the skin of hibernating bats, causing irritation that triggers repeated arousal from torpor. Each arousal burning fat reserves that the bat cannot replace until insects return in spring. Bats emerge emaciated, disoriented, and starving. The mortality rate in affected colonies was catastrophic. Little brown bat populations in the northeastern United States, once among the most abundant bat species on the continent, declined by over 90% in affected caves. Within 5 years of first exposure, the northern long-eared bat was listed as a threatened species within a decade of the disease's arrival. White-nose syndrome is now considered one of the most destructive wildlife diseases ever recorded in North America. The damage is not historical, it is ongoing. Surviving white-nose syndrome colonies showed behavioral changes, altered roosting positions, changed emergence, timing suggesting adaptive group-level response to a novel threat. In the years following white-nose syndrome's arrival, researchers monitoring surviving colonies documented behavioral shifts that were not random variation but appeared directionally consistent with reducing fungal exposure. Some colonies began roosting in cooler, drier cave sections, microhabitats, where fungal growth rates are slower. Others showed changes in hibernation depth and duration. Emergence timing shifted in some populations. Whether these changes represent genuine adaptive learning colonies, modifying their collective behavior in response to a recognized threat, or simply the differential survival of individuals who happen to prefer those microhabitats before the disease arrived, is a question that researchers are still carefully separating. The distinction matters profoundly. If surviving colonies are actively adapting changing behavior in response to experience, then the species has a mechanism for responding to novel threats faster than genetic selection alone would allow. If it is simply selection filtering the existing behavioral variation, the prospects are bleaker. The data so far is genuinely ambiguous. The bat's response to catastrophe is complex enough that we cannot yet fully read it. Bats practice social eavesdropping, listening to the echolocation calls of other bats to locate food sources they haven't found themselves. A bat hunting alone must locate its own prey, expending time and energy in systematic search before the echolocation can resolve anything worth pursuing. A bat that can extract location information from the calls of other successful hunters can shortcut that search, arriving at productive foraging areas without the cost of independent discovery. Studies have demonstrated that bats do exactly this. Individuals presented with playback of other bats feeding buzzes, the distinctive high-rate echolocation sequences that indicate a successful pursuit orient toward the source and fly to investigate. In natural settings, the spatial clustering of foraging bats around productive insect patches is partly attributable to this acoustic eavesdropping, each new arrival having used the calls of earlier arrivals as a location beacon. The information extracted is course direction and approximate distance, not precise coordinates, but it is enough to dramatically reduce search costs in an environment where productive patches are unpredictably distributed. The bat is not simply hunting, it is reading the landscape through the acoustic activity of its colony mates, building a distributed map of where the food is updated in real time by millions of simultaneous sensors. In some species experienced elder females lead colony foraging routes, knowledge of food locations transmitted socially, not instinctively. This was documented most rigorously in studies of greater spear-nosed bats in Trinidad, where researchers tracked individually identified colony members across foraging nights and found that movements were not random. Colonies followed consistent routes to productive feeding sites, and those routes were pioneered by older, experienced females who had visited those sites in previous seasons. Younger bats followed the elders. When elder females were experimentally removed from colonies, the foraging efficiency of the remaining group declined. The routes became less direct, the productive sites were reached later or missed entirely, and recovery of foraging efficiency required time proportional to the experience deficit created. The knowledge of where to go, built from seasons of individual experience, was being transmitted through social following. It was not in the genes, it was in the relationship between an old bat and the younger ones flying in her wake. Remove the elder, and the colony loses not just a member, it loses a library, one that took years to accumulate and cannot be reconstructed quickly, and was never written down anywhere except in the flight paths of an animal that will not live forever. The colony behaves as a distributed intelligence. No single bat decides when to emerge, yet millions take flight within minutes of each other, responding to light, temperature, and social cues simultaneously. There is no signal. No leader steps forward, no call goes out, no single individual makes the decision and communicates it downward through a hierarchy. And yet, across a cave containing millions of animals, the emergence happens coordinated, synchronous, timed with a precision that would be remarkable even in a species with centralized command. What produces this synchrony is a cascade of individual responses to shared environmental triggers. Light levels at the cave entrance drop below a threshold. Temperature shifts. The bats closest to the entrance begin to stir, producing movement and sound. Their neighbors respond to that movement. The cascade propagates inward and upward, each bat responding to its immediate social environment. Not to any global signal until the entire colony is in motion. The emergence is not organized, it self-organizes, and the distinction matters more than it might seem. A self-organizing system has no single point of failure, no leader to lose, no command channel to disrupt. 15 million individual decisions, each locally made, producing a colony-level behavior of extraordinary reliability. Evolution found this solution before the problem had a name. Some bat colonies occupy a single bridge and collectively consume more insects in one night than most people could imagine fitting into the sky above a single road. The Congress Avenue Bridge in Austin, Texas, houses a colony of approximately 1.5 million Mexican free-tailed bats beneath its concrete expansion joints, the largest urban bat colony in North America. Each evening between March and November, the colony emerges and disperses across the surrounding landscape, consuming an estimated 10 to 15 tons of insects per night. The bridge was built without any intention of providing bat habitat. The expansion joints happened to offer the precise dimensions of gap darkness and thermal stability that the bats required. They moved in. The city, after initial alarm, eventually embraced them. The emergence now draws thousands of human observers every evening in summer, people standing on the bridge or along the riverbanks watching the column rise. It has become one of Austin's defining natural spectacles. A concrete structure built for cars accidentally became the most productive bat maternity colony in an urban environment anywhere in the world. The bats did not ask permission. They simply found what they needed and moved in. Cave roosts occupied by bats for millennia develop unique microclimates shaped over generations by the collective presence of millions of bodies. A cave without bats is a stable passive environment, temperature determined by geothermal gradients, humidity by groundwater, airflow by passage geometry. A cave with a long-term bat colony is something different. Metabolic heat from millions of animals raises the temperature of roost areas measurably above ambient rock temperature. Respiratory water vapor from millions of breathing animals raises humidity. Ammonia from guano decomposition alters the atmospheric chemistry. The guano accumulation itself changes the physical substrate of the cave floor, creating new ecological niches, new drainage patterns, new thermal buffering. Over centuries, the cave is physically modified by its occupants, not through construction, as termites modify their mounds, but through the cumulative effect of biological presence at scale. The cave and the colony co-evolve. Each shapes the other. And when researchers enter a cave with a long bat occupation history, they are not simply entering a natural formation. They are entering a space that has been lived in, physically altered by life, for longer than most human cities have existed. When a roost is destroyed, the colony rarely reconstitutes. The collective knowledge of that location held socially dissolves with dispersal. Roost fidelity in bats is high. Individuals return to the same roost site year after year, and the colony's knowledge of that site, its microclimatic properties, its safety, its proximity to foraging areas, is embedded in the behavior of individuals who have used it across many seasons. When a roost is destroyed by development, cave gating, intentional extermination, or natural collapse, the dispersed individuals do not reliably find each other again. They scatter. Some find marginal alternative roosts. Many do not survive the transition. The colony as a social entity with its accumulated behavioral knowledge, its social bonds, its multi-generational roosting traditions ceases to exist. What is lost is not only the individuals, it is the social structure they comprised, the foraging knowledge distributed among them, the collective thermoregulation they provided to each other's pups. Restoration ecology can, in some cases, provide artificial roost structures that attract bats. But the colony that forms around a new structure is not the colony that was lost. It starts over. and the knowledge that took generations to accumulate must be rebuilt slowly from individual experience, one season at a time. Some researchers believe bat colony memory knowledge of roost sites, foraging routes, migration corridors represents a form of cultural inheritance found in very few non-human animals. Culture, in the biological sense, refers to information that is transmitted socially rather than genetically, knowledge, behaviors, and traditions that exist in the population rather than the genome, that can be lost when the social structure that carries them is disrupted, and that can evolve faster than genes because they are updated by experience rather than by selection across generations. By this definition, the evidence for bat culture is growing. Roost site knowledge transmitted through social following, foraging routes maintained by elder-led navigation, vocal dialects passed from mothers to pups, migration corridors used by populations for thousands of years and lost when those populations collapse. None of these are encoded in DNA. All of them exist in the relationships, the memories, and the behavioral traditions of living bats. The researchers making this argument are careful. Culture in bats, if it exists, is simpler and less elaborated than culture in great apes or cetaceans. But the architecture, social transmission, generational accumulation, vulnerability to social disruption appears to be the same. What the bat carries in its behavior may outlast what any individual bat carries in its genes. The loss of a single major roost can collapse insect regulation across hundreds of square kilometers, a ripple effect, invisible until crop damage appears seasons later. The lag between cause and consequence is what makes this so difficult to respond to. A roost is destroyed in spring. The insects that colony suppressed are not immediately more abundant. Populations take time to grow reproductive cycles, to complete generations, to accumulate. By summer, pest pressure is modestly elevated. By the following season, noticeably so. By the season after that, crop damage has increased measurably, but the connection to the roost destruction 2 years earlier is invisible to anyone not tracking both data streams simultaneously. This temporal gap between ecological disruption and visible consequence is one of the primary reasons that bat conservation struggles for policy traction. The harm is real, the accounting is delayed, and by the time the consequence is undeniable, the cause is historical, the roost is gone, the colony dispersed, the ecological function unrecoverable. Conservation biology calls this the extinction debt, the gap between when a disruption occurs and when its full consequences manifest. With bats, the debt is paid in insects, in crop yields, in pesticide costs, quietly, seasons later, without attribution. Bats in long-term study populations show individual personalities consistently bold or consistently cautious, a finding that has quietly unsettled assumptions about mammalian cognition at this scale. Personality in the scientific sense of consistent individual differences in behavioral tendencies across contexts and time was long considered a hallmark of cognitively complex animals. Large-brained mammals, primates, elephants, cetaceans. The emerging bat data complicates this picture. Long-term behavioral studies tracking individual bats across seasons have found that some individuals consistently approach novel objects, novel foods, and novel social situations more readily than others, and that this difference is stable over time across different contexts and not simply a product of age or body condition. Bold bats are consistently bold. Cautious bats are consistently cautious. The individual difference is real, it is persistent, and it has measurable fitness consequences. Bold individuals tend to discover new food sources faster, while cautious individuals tend to survive disturbance events more successfully. What it suggests uncomfortably is that the interior life of a small bat, the individual way it relates to its world, is more varied, more specific, and more persistent than our models of small mammal cognition have typically allowed. There is more in there than we assumed. There usually is. A bat colony in a single cave can maintain insect suppression across a radius of 100 kilometers, foraging ranges that cross state and county boundaries, invisible to any single jurisdiction's conservation planning. The Mexican free-tailed bat routinely forages up to 50 kilometers from its roost in a single night, sometimes further when prey distribution or wind conditions favor extended range. A colony of millions, dispersing across a circular foraging range of that radius, covers an area of roughly 7,800 square kilometers, larger than the state of Delaware, larger than many European countries. The insects suppressed across that range come from agricultural land, from residential areas, from wild land, from multiple political jurisdictions with different conservation policies, different pesticide regulations, and different attitudes toward bats. The colony does not recognize these boundaries. Its ecological function does not stop at county lines, but conservation policy almost always does roost protection. In one jurisdiction may be undermined by habitat destruction or pesticide use in another. Across a foraging range, no single agency has authority over. The bat's range is an argument for regional conservation planning that has not yet been sufficiently heard. To stand at the mouth of Bracken Cave at dusk and watch 15 million animals rise into the sky is to understand without words that intelligence does not require a single mind to hold it. After perhaps 30 minutes, the column rising from the cave entrance becomes so dense that individual bats are no longer distinguishable. Only the mass turning and dispersing a living river flowing upward and outward into the dimming sky. No individual bat is aware of the whole. No single animal sees the column from outside, perceives its scale, understands its structure. Each bat knows only its immediate neighbors, its own flight path, the echoes returning from the air around it. And yet the whole is coherent, organized, purposeful in aggregate without purpose at the individual level. This is what emergence looks like when local rules followed by millions of individuals simultaneously produce a global pattern that none of them intended and none of them can perceive. It is one of the deepest principles in biology. In physics, in the study of complex systems, and it is visible every evening between March and October at a cave entrance in the Texas Hill Country, if you know where to stand and are willing to wait for the light to go. Bats have been on Earth for over 50 million years. They were already ancient when the last mammoths walked, already established when the first human cities were being built. The oldest known bat fossil, Onychonycteris finnei, discovered in Wyoming and dated to approximately 52 million years before present, already shows the elongated finger bones and wing membrane attachment points of a fully formed flyer. The transition from ground-dwelling insectivore to airborne echolocator was already complete. Whatever intermediate forms existed, the protobats, the transitional ancestors, have left no clear fossil record. The bat arrived in the fossil record already fully bat, and it has remained in its essential architecture recognizably itself across the 52 million years since. Ice ages came, continents shifted, mass extinctions reshaped the biosphere. The bat was there for all of it, roosting in caves that have since been buried, hunting insects above forests that have long since become coal, navigating by stars that have shifted position in the sky. 50 million years of nights, 50 million years of echolocation pulses sent outward into the dark and returning with the shape of the world. And still going, tonight, outside your window, right now. In that 50 million years, bats survived every mass extinction event that reshaped life on this planet and emerged each time into a world that needed them more than before. The end Cretaceous extinction, the asteroid impact, 66 million years ago, that ended the dinosaurs, predates bats by roughly 14 million years. But the subsequent extinctions, the climatic collapses, the glacial cycles that drove countless lineages to oblivion, bats weathered all of them. The Eocene-Oligocene transition 34 million years ago was one of the most severe climate shifts of the Cenozoic. Global temperatures dropped sharply, tropical forests contracted, and many mammal lineages disappeared entirely. Bats survived. The Pleistocene glaciations, repeated cycles of ice advance and retreat over the last 2.6 million years. Compressed habitats eliminated food sources seasonally and drove many large mammals to extinction. Bats survived, retreating into caves, entering torpor, waiting. Each time the world rebuilt itself after catastrophe, bats were already there, already filling the nocturnal insectivore niche, already pollinating the recovering plants, already dispersing seeds into the recovering forest. They did not merely survive extinction events, they were part of what came after. Today, nearly a quarter of all bat species are classified as threatened or endangered, the fastest population decline in their 50-million-year history. The IUCN Red List currently classifies approximately 21% of bat species as threatened, vulnerable, endangered, or critically endangered, with population trends declining across a majority of assessed species. To place that in the context of the previous fact, bats survived asteroid impacts, glaciations, and continental rearrangements across 50 million years without a decline of this scope or speed. What is happening now is geologically instantaneous. Species that have persisted through every natural catastrophe the planet has produced in 50 million years are declining within decades. Within a single human lifetime, from causes that are entirely anthropogenic and entirely 19th preventable. The rate of change is the most alarming aspect. Evolutionary adaptation requires generations. Genetic response to selection pressure accumulates over centuries and millennia. What is happening to bat populations is happening faster than any biological mechanism for response can operate. The bat cannot evolve its way out of this. The only available solution is the one that created the problem in the first place: human choice. The primary drivers of bat decline are habitat loss, wind turbine collisions, pesticide reduction of insect prey, and direct hunting, each operating invisibly across different scales in different jurisdictions. No single cause is solely responsible. That is part of what makes the problem so difficult to address. Deforestation removes roosting trees and foraging habitat simultaneously, a double blow that neither protection of caves nor regulation of hunting can compensate for. Wind turbines kill bats through a mechanism called barotrauma. The rapid pressure change behind a passing turbine blade ruptures lung tissue without any physical contact, meaning bats can be killed by turbines they never touch. Pesticide use reduces insect abundance, not killing bats directly but starving them, reducing pup survival, and decreasing the reproductive output that sustains population levels. Hunting in parts of Southeast Asia and the Pacific removes individuals faster than slow reproducing species can replace them. Each cause in isolation might be manageable, together operating simultaneously across the bat's entire range. They constitute a pressure that has no historical precedent in 50 million years of bat existence. This threat does not recede seasonally. It does not spare the cave. Wind turbines kill an estimated 600,000 to 900,000 bats per year in North America alone, a death toll accumulating in silence, largely uncounted and largely unaddressed. The barotrauma mechanism was not understood initially. Early mortality estimates focused on direct blade strikes and substantially undercounted total deaths, because barotrauma victims often show no external injuries and are not identified as turbine casualties in standard monitoring. Revised estimates incorporating barotrauma mortality elevated the figures dramatically: 600,000 to 900,000 per year in North America. Globally, the number is unknown. Wind energy infrastructure is expanding rapidly across bat habitat in Europe, Asia, Latin America, and Africa, with mortality monitoring that ranges from inconsistent to non-existent. The species most affected, the hoary bat, the eastern red bat, the silver-haired bat, are migratory tree-roosting species that concentrate along ridgelines where wind energy development is most intense. They are also among the least studied, least monitored, and least protected bat species in North America. The turbines run at night. The bats migrate at night. The deaths accumulate in the dark, counted imprecisely, reported inconsistently, and addressed, so far, inadequately. The economic value of bat, pest control, pollination, and seed dispersal is estimated at over $50 billion annually. Worldwide ecosystem services that carry no invoice and appear in no national account. $50 billion per year, delivered by animals that most people fear, many governments do not protect, and essentially no one pays for. The figure comes from peer-reviewed economic analyses attempting to monetize the ecosystem services provided by bats across their global range, a methodology that is imperfect by definition, because converting biological function into currency requires assumptions that can always be contested. But even the contested figure is instructive. It tells us something about the gap between the value bats provide and the consideration they receive in land use decisions, in conservation funding, allocations in the design of wind energy, projects in the regulation of cave. Disturbance. When a developer proposes to destroy a bat roost to build a parking structure, the economic analysis typically does not include the value of the pest suppression service that roost was providing to the agricultural land within a 50-kilometer radius. It does not appear in the cost-benefit calculation. It does not exist in the accounting until it is gone, and by then the calculation has already been completed, the permit issued, the roost demolished, and the debt quietly added to a ledger that no one is officially keeping. In their absence, the insects they suppress would devastate grain crops, the plants they pollinate would fail to reproduce, and the forests they seed would thin and fragment across decades. This is not a prediction. It is a description of what has already been documented in areas where bat populations have collapsed. In regions of North America severely affected by white nose syndrome, studies have found measurable increases in agricultural pest moth populations in the years following colony collapse, and corresponding increases in insecticide application rates on nearby farmland. In islands where flying fox populations have been hunted to extinction, researchers have documented declines in the regeneration rates of forest tree species that depended on those bats for pollination and seed dispersal. The forest does not disappear immediately. It simplifies, loses species, loses the diversity of its canopy, becomes less resilient to the next disturbance. The process is slow enough to be invisible within a human attention span, and fast enough to be irreversible within a human lifetime. The bat holds things together at a timescale that is longer than we tend to think about, and shorter than we tend to act on. That gap between the timescale of ecological consequence and the timescale of human attention is where most of the damage gets done. Cultures across Asia and parts of Africa have long regarded bats as symbols of luck, longevity, and good fortune. It is largely Western tradition that casts them as creatures of darkness and dread. In Chinese culture, the bat fu shares its spoken syllable with the word for good fortune, and has been a symbol of prosperity and long life for over 2,000 years. 5 bats depicted together, Wu Fu, represent the 5 blessings of health, wealth, longevity, virtue, and a peaceful death. Bat imagery appears on imperial robes, on porcelain, on jade carvings. On architectural ornament, a consistent presence across millennia of Chinese decorative tradition, always auspicious, always welcome. In West African traditions, bats are associated with spiritual perception, the ability to navigate darkness, being read as a form of wisdom. In pre-Columbian Mesoamerica, the bat deity Camazotz was a figure of power and transformation, feared but also respected. The Western association of bats with vampires, with disease, with Halloween darkness is culturally specific, historically recent, and demonstrably at odds with both the biology of bats and the majority of human cultural traditions that have existed alongside them. Fear in this case is not universal. It is a choice, and like most choices, it can be revised. Scientists still debate the full origin of bat ecophobia in Western culture, whether it traces to medieval plague associations, vampire folklore, or something older and harder to name. The plague hypothesis suggests that bats emerging at dusk in large numbers from the same caves and cellars where rats congregated became unconsciously associated with the disease and death that rats carried, even though bats had no causal role in plague transmission. The vampire folklore hypothesis traces to 18th century European encounters with accounts of the Latin American vampire bat, misunderstood, exaggerated, grafted onto pre-existing Eastern European undead mythology to produce the literary vampire, which then retroactively darkened the reputation of all bats by association. The older hypothesis favored by some evolutionary psychologists suggests that a general wariness of small, fast-moving, unpredictable creatures in low light may be a deeply embedded perceptual bias, a legacy of a mammalian past in which such stimuli genuinely warranted caution. Perhaps all three contribute. Perhaps none fully explains the specific intensity of bat aversion in Western contexts. What is clear is that the fear is cultural, not biological. It was constructed gradually from misunderstanding, layered on misunderstanding. The bat paid for it. Still is. The bat is one of the only animals on Earth that has been simultaneously essential to human agriculture, instrumental to human medicine, and persistently misrepresented by human culture for the entirety of human civilization. Pest suppression that has protected grain harvests for 10,000 years of agriculture, delivered nightly, silently, without acknowledgement. Compounds in bat saliva, now informing stroke treatment. Immune system biology reshaping our understanding of viral disease. Wing biomechanics informing drone design. The bat has been contributing to human welfare across domains of medicine, agriculture, engineering, and ecology simultaneously, and has received in return extermination campaigns, habitat destruction, deliberate cave disturbance, and a starring role in the iconography of horror. The relationship is not balanced. It has never been balanced. The bat gives. We take the benefits, ignore the source, and fear the provider. It is one of the more consequential misunderstandings in the history of human relationships with the natural world, and it is still ongoing tonight in caves and bridges and tree hollows on every continent where bats and humans share the dark. The bat asks nothing of us. It emerges after dark, does its work in silence, and returns before dawn, and the world it quietly holds together has never once thought to say thank you. There is a philosophy embedded in the bat's existence that has nothing to do with philosophy. It simply does what it does completely every night, without recognition and without request. It does not need our approval to pollinate the baobab. It does not need our gratitude to suppress the moth that would otherwise consume the grain. It navigated the dark for 50 million years before we arrived, and it will, if we allow it, navigate the dark long after our cities have grown quiet. What the bat asks of us is not gratitude. It does not ask anything. What it requires, the only thing it requires, is the continued existence of the world it evolved within. The caves intact, the forests present, the insects abundant enough to hunt, the nights dark enough to navigate. They are simply the world, as it was the world the bat has always known, and the world we have been, with increasing speed and thoroughness, remaking into something it does not recognize and cannot survive. The bat does not ask us to save it. It simply continues, doing what it has always done. In the dark,